Okay, what's happening next Wednesday?
Pizza Day.
Pizza Day, yeah. Actually, I did win pizza for my CLE bike to work team. So if you had joined, you'd be getting free pizza. But
Midterm number 2.
Midterm number 2, exactly. And that means that Monday will just be review on all the stuff that we've done so far. I'm not planning on doing clicker questions that day, so I expect that no one will be here. Well, we shall see. But bring lots of questions as far as that's concerned. Just really briefly to review what we talked about last time, these wonderful filoviruses that everybody gets really scared about, at least in terms of these surveys that we talked about last time. The virus we're going to talk about today is way scarier, but nobody seems to worry about it as much. But yeah, basically, again, take-home message as far as these filoviruses are concerned is they're basically just really big rhabdoviruses, with those couple of little exceptions. The VP30, which is regulating the amount of messenger RNA production. I hate the transcriptional regulation thing. And we have that secreted glycoprotein as opposed to the normal, what do you call it, sausage? Noodle, yes, that we had last time. But otherwise, again, extremely similar. You've got stuttering, you've got the start-stop kind of way to make subgenomic RNAs, et cetera. And the one thing that we didn't get to last time that I did want to mention just really briefly is the ecology of what people think is going on with Ebola virus. It's not entirely clear, all of these different things here, but the basic idea, and I also wanted to bring this up, the enzootic cycle, which is probably where the reservoir species are, is circulating probably in bats and generally doesn't seem to cause them much in the way of disease. So classic kind of reservoir species. May be transmitted to a different species of bat. We really have no idea. This then gets transmitted to non-human primates. And interestingly enough, this small deer species also seems to get infected. And both of these get rather sick. And then the next question is, what about humans? Could be from bushmeat, could be directly from the bats. And the big issue with a real human epidemic is that it will actually spread between people. And this is unusual for most of these zoonotic diseases. Very often the human is really a dead-end host, so humans can't actually transmit disease. Ebola is a bit of a difference as far as it's concerned. There's lots of really good information at the CDC about all of these filoviruses. Yes?
I noticed that you talked about bats as a reservoir a lot, and I was wondering if there's ever been a case, I don't think people would ever really notice this, as humans being a reservoir for a disease that infects animals.
So the question is basically, bats seem to be this reservoir, and exactly why that is also not clear, which you didn't ask, but about humans being a reservoir for other animals getting sick. And the answer to that is most people thought that wasn't true until recently, actually, as far as flu is concerned. It does look as if potentially the swine flu that happened in 2009 actually may have gone from humans to pigs. So there's a definite possibility that some of these, definitely is a lot more back and forth happening. So these arrows are certainly not just going towards the humans. There are certainly some cases where it seems to be going the other way as well. Not that us anthropomorphic types have a tendency to think about it that much, but it's a really important point, and I think people are just now beginning to think about that. Okay, more questions, comments, worries about filoviruses. There's a current outbreak in the Democratic Republic of Congo. Some of you may have seen that. I think something like 7 deaths so far. Nothing that major yet. Nicole?
Can the virus go through into the feces of the bats, and that's how it's mainly transmitted? A lot of those places they use one.
So Nicole's question is basically, well, I'll paraphrase here. Is it bat guano, or basically, what is this blue line, really, in terms of how it's being transmitted? Nobody really knows. And that's been one of the big questions. As we talked about last time, a lot of these filovirus virions are actually pretty wimpy. It's hard to get infected by them unless they're really, really, really large numbers. So guano seems unlikely, but nobody really knows at this point. Okay, other questions on this? And for a big surprise, no clicker question. There'll be some later, don't worry. So big picture on these orthomyxoviruses. Practically nobody says orthomyxovirus. It's the only one of the only kind of orthomyxoviruses that we know of are the flu viruses. So everyone talks about flu or influenza. So that'll be all I will say about orthomyxoviruses. I'll pretty much always talk about flu or influenza. These are now, instead of being mononegavirales, these are segmented genomes. And we won't talk about any of the other segmented RNA virus genomes. Someone asked me about hantaviruses after class yesterday and 2 days ago. And those are also segmented negative strand viruses. And there are a few others as well. But again, we're only just going to talk about flu as far as this is concerned. So what are the big concepts here? Think about these when you're thinking about the exam. Segmented genome, again, we haven't really talked about segmented genomes much except for, who's my next victim? Jurikits. Yeah. What are the segmented genomes that we talked about? Can you remember any? Way back when, right after the last midterm? So we talked about lambda, we talked about the plant viruses, we've talked about picoRNA viruses, we've talked about flaviviruses, we've talked about coronaviruses, we've talked about the filoviruses and the paramyxo and rhabdoviruses. Any of those have segmented genomes? You remember any that do? I've been told one thing that I should do is allow people to pass, and then I come back and ask them the next time around. So I'll probably start doing that next year. Any other ideas? I know. I'm also thinking about having a course evaluation at the time of the midterm, an anonymous one, so you can let me know what you think about this whole roll call business. Yeah, you'll let me know. I figured as much. I trust you to do that. So, any other thoughts? Yes? Pardon? Cucumber mosaic virus and a lot of other plant viruses as well are those with segmented genomes. And that's one of the neat things about those segmented genomes, of course, with the plant viruses. They even have different capsids, which is also just, at least for me, kind of mind-blowing. Hemagglutinin, we've talked about hemagglutinin a whole bunch, but there's been really nice studies on hemagglutinin, and we'll revisit it a little bit today, because the best known of any of the fusion proteins, I would argue, is the hemagglutinin from flu. Neuraminidase we've talked about a little bit. I'm going to spend a little bit more time talking about this today. The neuraminidase cleaves off the receptor, which is sialic acid, for hemagglutinin, probably to make sure the virus can be released from the cells that it's infecting. And you wouldn't think that this is a really critical aspect of the viruses. Turns out that almost all of the anti-flu medicines that work are all anti-neuraminidase proteins. And I say small molecules, I should say. Matrix protein, we talked about this a little bit before, particularly having to do with its activity as an ion channel. So changing not only the pH on the outside of the virion, but also changing the pH on the inside of the virion. One thing which is very bizarre about these viruses is they replicate in the nucleus. But wait a minute, it's an RNA virus, Dr. Stedman. What the heck are they doing replicating in the nucleus? We'll get back to that. The main reason is that they snatch caps, and we'll talk about cap snatching a little bit later on. So as usual, orthomyxoviruses, what do you mean by orthomyxoviruses? We just mean flu. talk a little bit about where they were found. I'll leave the disease stuff off until the end here. Structure, binding, and entry, and then big interesting things here relative to everything else that we've talked about have to do with messenger RNA production and replication. And this switch between those two processes, and then release is pretty standard. Actually looks a lot like the paramyxone rhabdoviruses, and if you look carefully, the RNP that I was giving people a hard time about last time is actually from that particular same slide here. And then again, we'll talk about disease, which is way scarier. At least it should be way scarier than Ebola. This is one of my favorite electron micrographs that I've ever seen. This is the citation down here. This is a flu virion, and the thing that I really like about this is you can actually see in this micrograph all of those segmented parts of the genome all the way through here. It's really an absolutely incredible electron micrograph. So where or what are the orthomyxoviruses? Flu. There's A, B, and C. And one very important thing, and this is what unfortunately lots of people say, and probably why people aren't as scared of influenza as they should be, is that this sniffle I have is the flu. What I have a stomach bug, it's the flu. No. The rhinovirus and coronaviruses are what's causing the common cold, and flu is much worse than that. And in fact, you can die from flu. The coronaviruses, you can die from SARS, but the vast majority of them you won't. And rhinoviruses, I don't know anyone who has died from a rhinovirus infection. So what do these look like? They're enveloped viruses. This envelope gets picked up at the plasma membrane. So these bud off of cells. Here's an electron micrograph of some of these virions budding off of the cell. Depending on whether it's flu A, B, or C, it goes from 6 to 8 helical nucleocapsids. We're just going to talk about flu A, which has 8 of these helical nucleocapsids. And those are what's inside the membrane. And again, we go back to here. That's, if you count very carefully, 1, 2, 3, 4, 5, 6, 7, 8 of these segments packaged in one virion. You'll also notice that it's pretty pleomorphic. When I say pleomorphic, having lots of different shapes. And so some can be those almost spherical form, as we saw before, but often there will also be these more filamentous-like virions which are being produced. So what do these look like in cartoon form? We've got these 8 subgenomic, or subgenomic, I shouldn't say subgenomic, these are segments of the genome that are packaged inside, and again, this nice sphere here, almost never you're going to see a nice spherical one of these viruses because there's nothing that's really holding it in this sphere form. And we'll talk much more about these guys later on. These 8 genome segments encode 10 proteins. That tells you immediately that at least, well, one of them could be making 3, but in fact 2 of them make 2 different proteins. And 9 of those proteins are actually packaged. So it's basically almost all of the proteins that are made by each of these genome segments gets put into the final virion. So let's look at those proteins. We've already talked quite a bit about hemagglutinin, and we'll talk more about that. That's your receptor binding protein. The neuraminidase, which is also an envelope protein, that neuraminidase cuts off this receptor. Then we also have this ion channel, the M2 protein, which allows the pH to change on the inside of the virion. Then we have a much more normal, quote unquote, we've talked about these a bunch before,